Welcome to our review on alpha, beta and gamma radiation. First thing we need to understand then is what we're talking about when we refer to radioactive decay. Now, what we find is that the nuclei of some atoms are unstable. So what then happens is, because they're unstable, those unstable nuclei are going to break down, and as they do this, they emit radiation. So that is the process of radioactive decay, and that's how we get this radiation being released. What we find is that any of this radioactive waste we mentioned when talking about nuclear reactors, it's actually dangerous because it contains a high concentration of these radioactive atoms, so these unstable nuclei, which means that that radioactive waste is going to be undergoing this process of radioactive decay and therefore emitting radiation. So there are three types of radiation that we need to remember. We've got alpha particles, beta particles, and gamma rays. Now we can actually use this bit of equipment called a Geiger-Müller or GM tube to detect any form of nuclear radiation. Now what we find is that when we use our GM tube and we set up experiments using different objects to see what can pass through it, then the different types of radiation can penetrate materials to varying extents. So that diagram that I've given you is important to remember. So if we look at the top one there, our alpha particle first of all, then we can see that's the least penetrating. So literally we can stop alpha particles with a few sheets of paper. So that one is very easy to stop. The beta particle is our next most penetrating, if you like, and this one will be stopped by a few millimeters of aluminium. And finally, our gamma ray, this is the most penetrating of all three. So the only way we can stop our gamma ray is with actually using meters worth of concrete or several centimeters of lead, and even in those cases there is the risk that some can still get through it. So gamma is the most penetrating, alpha is the least penetrating. And remember, we can actually use the degree of penetration then to work out what type of radiation is present. So if we've got an unknown radiation source, we can put some bits of paper in front of it. If we do not pick up any radiation on the other side, it was obviously alpha. If we do, it could be beta or gamma. Whereas if we only detect radiation when we put it through some aluminium, for example, then we know it's got to be gamma because beta and alpha are stopped by them. So look at the question if they give you one to talk about what type of radiation is present. Look and see what it's able to penetrate through. If it's able to penetrate through the paper, the aluminium, then we know we've got our gamma. If it's only able to penetrate the paper, then we've got beta. If it doesn't penetrate through anything there, then we have our alpha. So make sure that you look at the information in the table and remember the order of the penetration. So alpha, beta and gamma are all types of ionizing radiation. So what this actually means is that when that radiation comes into contact with atoms, it causes either the addition or removal of electrons from that atom. So as a result of that, our atom becomes an ion, a charged particle. Now, if the electrons are being removed, then we're going to make a positive ion, whereas if we're going to add electrons to it, it becomes a negative ion. So we have a pattern again to remember here in terms of which is the most ionizing. The alpha particles are the most ionizing, and our gamma rays are the least ionizing. So it's the opposite of their penetration power. So the most ionizing is alpha, the least ionizing is gamma. If this ionizing radiation actually comes into contact with our human cells, and therefore we're ionizing atoms within those cells, then what we can see is either damage or even death of that cell. So if we look at the DNA that could become ionized within the cell, then what's going to happen there is it's going to change how it interacts. So what we'll find is the ionized parts are either going to attract or repel each other, so that means that it's going to change the way the cell behaves because the DNA isn't behaving as it was. We can also see the effect of chemical reactions that wouldn't normally be taking place being initialized as well. So if we actually have very severe damage to the DNA, then that can lead to one of two things. It could lead to cell death outright, or it could lead to the development of cancers. Last thing we need to consider is how we actually handle these radioactive sources safely. First thing is that whatever we're storing them in must be appropriately labelled with the radioactive sign and what's actually contained. anytime we are handling it, we should be wearing gloves. If we're talking about the more penetrating radiations, then really we should be standing behind a lead shield or wearing a lead apron. You should always minimise the time that you're exposed to these radiations and always keep your distance. So as opposed to picking it up with your hands, you should use tongs to handle the sources so it's not coming into close contact with your skin at all.